This lecture will cover the gram-negative bacilli identification. So this will be the organisms that grow on the MacConkey agar. And of course, some of these tests have already been, or some of the tests that would be used to identify these organisms have already been covered with the gram-positive lab lecture. So it may be useful for you to refer back to that particular lab and to keep those printouts handy from here going forward for any of the tests that we'll cover coming up with some of these different organisms, because it will utilize some of the same tests. So our Enterobacteriaceae cultivation is usually consistent of just routine agar with 5% sheep blood agar, MacConkey agar. Depending on the particular specimen that you're culturing, it could also include a chocolate agar. And then if you're doing something like a stool culture, or you're looking for any sort of particular enteric bacteria or cause for any sort of issues with the stool culture, then there are other agars that can be thrown in. And so an example of this would be some labs would choose to do an HE or an XLD agar, maybe a Salmonella Shigella agar, and MacConkey sorbitol if they're looking for EHIC. Some of your enrichment broths that may be also used would be like a gram-negative broth or an SEL broth. With Pseudomonas aeruginosa, it is able to grow at 42 degrees Celsius, although it's not typically going to be set up to be cultured at 42 degrees. Again, this will grow on your routine agar, so usually it will be set up on blood and MacConkey agar. And then, of course, if you were, depending on the lab that you were working at, if you were trying to determine any other particular characteristics of it, then you would also use something like a Mueller-Hinton. So Mueller-Hinton agar is actually used to do antimicrobial sensitivity with those discs. So you see the antibiotic disc up here that are placed onto the agar. So there's a special technique that the lab will use for these discs when determining if an antibiotic will work for a particular infection. And so most labs do this automated now, but there are still some labs that do a manual method, especially with some of the rare antibiotics or older antibiotics. And so if it were plated on the Mueller-Hinton plate, then you could start seeing some of these characteristics from that diffusible pigment. And so where you get those different shades of color, and here's like that blue color that I was talking about earlier, the green color, and sometimes it kind of gets like a little fluorescent look to it, the red color, and of course there is a browner pigment that kind of makes a brownish darkest type color to that agar. Couldn't find a good picture of that. So with most labs that choose to set up an HE or an XLD agar, they're looking as like an early screen to see if they can determine some of these enteric pathogens and what they are. So while most of them will present with a yellow or salmon type color to it, you do have your Salmonella that will have those black centers from that H2S production, and your Shigella will be just a green colony with no H2S production. Your XLD colonies would present as a yellow for most organisms. So deep red color with that black center for that H2S production. And then and so this would be your Salmonella. Your Shigella would be clear, colorless, so it might just kind of appear as a red colony, but it's actually, you can just kind of see through it. Isolation of your Vibrio species can be done through routine culture media, such as your sheep blood agar or your MacConkey agar. And one thing that's important to note, some of you may have noticed that already work in the healthcare setting, that stool samples are submitted in a couple of different collection medias. So you may have the OMP or the ovum parasite collection containers, as well as your culture and sensitivity collection containers, and those have a little bit of liquid inside of them. And so that liquid media is actually to help preserve the organism um, that are in that stool sample, as well as to keep anything from overpopulating the other organisms. But one important thing to note is that with your Vibrio species, you're looking for stool samples to be collected in the Carey-Blair media, because the buffered glycerol saline is very toxic to the Vibrio strands, and so that's why you do see different collection containers for one stool culture, and they will actually submit a sample that is not preserved in anything, so it would just be like in a urine cup. Stool is also plated on a TSB agar, and what this will do will look for the organism's ability to ferment sucrose, which would give it that yellow color, so you see in that second image, as well as the green color would indicate a non-sucrose fermenter. So this can help differentiate the different Vibrio species. And then there's also chromogenic type of Vibrio agar that can help to differentiate them, and that would be based on the particular growth pattern that it has, or the color that it has. So you can see these organisms over here to the side where you've got some blue and pink type growth patterns. So just to recap this flow chart, when we look at growth on MacConkey agar, we're thinking about a gram-negative bacilli, and we're determining if it's a lactose fermenter or a non-lactose fermenter. And so from there, we would do an oxidase test to determine if we're dealing with some of our Enterobacteriaceae or non-Enterobacteriaceae. And so with our oxidase test, we did cover this within our gram-positive lecture, or within our gram-positive lab lecture. And so what we're going to do next is our indole test. And so we're going to discuss that test first, and then we'll work our way through some of these other tests. So our spot indole, there are two different methods. Our spot indole is a rapid method. And so with this, what we're looking for is the presence of this tryptonase. And so if it is present within that organism, when we put usually some sort of type of filter paper or something down, we put the reagent on it, and then we take a little bit of that organism, and then we place that onto that filter paper. And then if it's positive, we get that blue reaction. And then if it's negative, you have no reaction. And again, sometimes, like some of our other tests that we've discussed before, you can see a little bit of an outline of that organism, and that's okay, but it didn't turn positive. And so if we were using a media to do our indole tests, broth media, then that reagent would be added to the top of that inoculation. So that organism has been put down into that broth, and then we add the indicator reagent to the top of it, and what we're looking for is that pinkish color or reddish color for that positive test, and no color change for our negative test. For a citrate test, we're looking for the organism's ability to use the sodium citrate. And so what will happen with that, if the organism is able to grow in this media and use that citrate, then you'll get a color change, and what you're looking for is this blue color here. And you can see this organism is still a little greenish color here at the bottom, but you're looking for that blue color change to be a positive reaction versus no change for your negative reaction. With your oxidation and fermentation, what you're looking for is to see if these organisms can if they're able to either oxidize or ferment different carbohydrate substances. And so depending on which one you're looking for, you would use that particular carbohydrate. So for example, we've mentioned with some of our non-Enterobacteriaceae, their ability to ferment glucose. Of course, most of your Enterobacteriaceae do ferment glucose. So what you're looking for is you would actually take a particular medium that has that particular carbohydrate or sugar inside of that media. And you would take 2 of those tubes and inoculate both tubes with several stabs. So you can see here up top where these organisms have been inoculated with several stabs into that media. And so you can see where you have those little stab marks. And so once that organism is placed into that, then is incubated for up to 7 days. And of course you're looking for any sort of production of acid within that tube. So you would do 2 tubes for each particular sugar that you're trying to test for. So you can see that these 2 tubes here, both of them are labeled A, where you have one tube that has no oil laid on top, and then you have one that does have like a little oil layer on top. And so what that oil layer does, that mineral oil, will actually create an anaerobic environment. And so if there is any sort of color change, like an acid production, then it will produce a yellow color change. And so what our results would indicate is if we're having an acid production that's indicated by that color change, then we know that our organism is able to ferment that particular sugar. However, on the other hand, if it is an oxidizer, it will only be in the presence of oxygen. So here at B, you can see this tube here is an oxidizer because it had oxygen here, and this one had no oxygen. That mineral oil was on top. And so the fact that it needed oxygen in order to oxidize that sugar made it an oxidizer. Whereas in tube A, it didn't matter if oxygen was present or absent, it is still able to ferment that glucose in the presence and without the presence of oxygen. And whereas tubes C, neither one of them had any sort of change with or without the presence of oxygen. So it does not utilize any of the sugar that was in that tube. So these tubes could have all been glucose, or they all could have been sucrose. You would basically test the particular sugar that you're looking for. And a lot of the modern methods today either use analyzers or they will use a long strip test where the sugars are all actually within little wells within that strip, and you'll load the organism into each individual one. So if, for example, if you're looking for glucose in one, lactose in one, then you're looking for the color change in each of those. So maybe it's yellow in these two, so you know that it will ferment glucose and lactose for both of those tubes. And again, some of these have the same way of looking for oxidation, where oil is actually laid over top of those little wells to see if it will oxidize that sugar as well. The DNase test is looking for an organism's ability to produce the deoxyribonuclease. And so there's actually a video that's loaded in the lab section that shows this test, but the method is a little bit different. But he does demonstrate what you're actually looking for. And so with this particular test, when it's set up on this particular media, what you're looking for is the DNA to actually be hydrolyzed. And so what that will do is actually create a clearing of that media around that organism. And so if there is a clearing around it, that is considered positive. If there's no clearing, that is negative. The urease test is used to determine the organism's ability to produce the urease enzyme, which is used to hydrolyze urea. And so with this one, it does produce a color change once those products are broken down into ammonia and CO2, and then you can get a color indication. And if the agar slant was to turn a magenta or pink color or a light orange color, then that's considered positive, and of course no change is negative. The OMPG test looks for the ability of the organism to produce the beta-galactosidase. With this particular test, it helps us to determine a late lactose fermenter from the non-lactose fermenters. We did mention that there were a couple of the Enterobacteriaceae that were the late lactose fermenters. So this would be a useful test for that. If the organism is positive, it would produce a yellowish, orangish type color. And if it was negative, it would be colorless. So here you would see a positive result versus a negative result. The decarboxylase test differentiates the different Enterobacteriaceae from other gram-negative rods. And so what you're looking for is the ability of the organisms to hydrolyze some of the different amino acids. So those amino acids would be the arginine, lysine, and ornithine. And so what you would do is you would put, all of those would be in a different tube, and you would inoculate that organism in it. And what you're looking for is it to hydrolyze those different amino acids, and then it would result in a color change due to a pH indicator within those tubes. So this particular method here would be looking for that purplish reddish color to be a positive result, and then no change would be a negative result. So this last tube here is an uninoculated or no change tube. So that particular tube, you can see that the color is pretty similar to the negative. So this negative tube, this organism was not able to hydrolyze that particular amino acid. The lysine iron agar is used to differentiate gram-negative bacilli based on their ability of decarboxylation or deamination of the lysine, and their ability to form the hydrogen sulfide or the H2S. So this agar is made into a slant. It actually contains the lysine within the agar. And so with this slant, you've created an aerobic environment up top, and then the bottom is an anaerobic environment. So when you introduce the organism, you would stab the slant, and then you would also streak the slant. So you would stab down into the agar to the bottom and streak the slant. And so you're looking to see how this organism will act in the presence of the lysine with and without oxygen. If the organism is able to ferment glucose, then it will actually start to create an acidic environment at the bottom, and it will turn yellow. If the organism is able to produce the lysine decarboxylase, then it will actually form cadaverine, and that will actually neutralize the organic acids that happens with the glucose fermentation. And this means that the butt of the medium will actually go back to an alkaline state. So it will actually change that color back to that dark purplish type magenta looking color. I don't have a color to match, so I'll actually change it back. And then from there, if the decarboxylase is not produced, it does not produce that cadaverine, which means it will stay that acid yellow color. But if it does, then you'll get that purple color, or will revert back. If the oxidative deamination of lysine occurs, then a compound is formed that will, in the presence of that ferric ammonium, and a coenzyme, and then what it will do will actually produce a burgundy color to that slant, and so that slant will actually change color as well, and that's because you do have the aerobic environment up top to be able to allow it to do that. If the deamination does not happen, then the slant will actually stay purple. So here into the top corner, here's some images of some of those different things that have happened. So with your very first slant, you have an alkaline environment. So you have the lysine decarboxylation that has happened, but you do not have any sort of fermentation of the glucose. Next, you have a alkaline, and let's see, alkaline and probably an alkaline, but it does have the H2S production, so it has turned that agar black. And then our next slant, we do have an alkaline. You can see that little bit of magenta color up top with an acidic. So there at the bottom, that has turned to an acidic. So it has actually fermented the glucose. And then with a D, this one has actually turned red, and then you have an acidic butt. With that, it has lysine deamination, and it has glucose fermentation as well. And then this last tube is no change, it's not inoculated. So this would be what the tube would look like when you start. Triple sugar iron agar is used to detect if a gram-negative rod can ferment glucose and lactose or sucrose, and also whether or not it can form the H2S. And so this is used to differentiate the different members of the Enterobacteriaceae from other gram-negative rods that can be found within your specimens. And so this particular agar contains lactose, sucrose, and glucose, as well as some indicators. And if glucose is the only sugar that's fermented, then there will be a small amount of acid production that is produced on the slant that will be oxidized to a more neutral product. So it'll go from an acid appearance, which would be yellow, and then it would actually go back to a red appearance, just where it would be in the presence of the oxygen, and it would remain at an alkaline type state. Whereas within the butt, there's no oxidation, so it would not revert back, so that butt would remain yellow. If lactose and sucrose are fermented, then there would be like an overproduction of the acid, so the slant as well as the butt would all turn yellow. So here at the top you have an uninoculated tube in tube D. So you can see how the tube would start, so it'd be pretty obvious if that tube color did change. Then from there, if the specimen was alkaline, or if there was no change in the slant or in the butt of the tube, then you have a non-fermenter of glucose, lactose, and sucrose. And so that particular presentation would be here in C, where you have an alkaline. so you don't see any sort of color change. An alkaline acid, so you have an alkaline slant, acid but. This would indicate glucose fermentation only. So you would see this in this second tube, tube B, where you have alkaline acid. So you have just the glucose fermentation here, not at the top. You can see a little bit right here on that slant where that is still alkaline, but you have the acid at the bottom. And of course this one has that H2S production because you have that blackening there. And then if we're looking for an acid-acid, so of course this used all three. So you get that acid production in the bottom as well as on the slant. And then you can also see where this particular organism produced gas. So you have a break in that agar. Depending on the organism, sometimes it will actually just kind of shred this agar and it'll be broken up all the way through. But most of the time you'll just have a little bit. Sometimes you can have an agar slant that will, excuse me, it'll actually push up from the bottom. So there'll be like a little layer of air at the bottom, and then all of the agar would be through here. So if you were going to stereotype any of the organisms to determine what particular virulence they have, then you would actually use a serotyping kit to do this. And so these are more often used for your Salmonella, Shigella, and E. coli. With your Salmonella, you're looking for antisera A through E, which is looking for those O antigens. And then you're also looking for that capsule, that K antigen. And so a lot of times, depending on the particular lab, you may see this more in reference labs than you would just a hospital lab. Because this would be so specialized, you wouldn't really see this that often to be able to need to stereotype these. So this may be something that would be sent to like the CDC or the state lab. Your Shigella, again, you're looking for some of the different somatic antigens. And then of course with your EHIC, your E. coli 0157H7, if it were to be suspect on your sorbitol agar, so it did give those colorless type colonies on the sorbitol, then you would want to do serotyping on that to confirm that that is what that organism is. Your nitrate reduction test determines whether an organism can produce nitrites, and so microorganisms that are able to reduce nitrite to nitrogen will not turn any particular color, but it will produce a gas in the nitrate reduction. And so this is due to nitrates being converted to nitrites. And what you're looking for is a colorless gas or a colorless broth media with gas production. So you can see that little gas bubble there. This will be considered positive. And then of course this is negative. The string test is used to differentiate the Vibrio species from Eriomonas. So with this test, you would actually emulsify the organism into a sodium dioxalate type of reagent. And then what this will actually do, it will lyse the Vibrio cells, and that lysis will result in the release of DNA. And then that DNA can actually be pulled up kind of like a string. You can actually see it with any sort of like stick or inoculation loop. And so to be positive, it would actually have to form a string. A negative would be no string. If you ever did any sort of like DNA lab in high school, you may have done something like this with strawberries or saliva, where you're trying to do a DNA extraction. And so this would be sort of similar to that particular test method.